Welcome to Curiosity, the podcast where science meets wonder. Join us as we explore the fascinating, the innovative, and the unexpected, from the secrets of the cosmos to the marvels of everyday life. Each episode takes you on a journey to uncover the knowledge and stories that fuel our endless quest for understanding. Let your curiosity guide you.
I was sitting with this stack of papers last night, the ones we're covering today, and I couldn't help but think about the second law of thermodynamics.
Entropy.
Entropy, exactly. The idea that everything just inevitably moves from order to disorder.
The coffee gets cold.
Right, the coffee gets cold, the mountain erodes into sand, the star burns out.
Yeah.
And for the entirety of human history, we've viewed our own bodies through that exact same lens.
That we're just complex machines, but machines that are ultimately subject to that universal law of decay.
Yes. You're born, you develop, you peak, and then the slow, grinding slide into dissolution begins. It has always felt like a physical law,
Yeah.
as unbreakable as gravity.
It's the one narrative that every single culture shares, really. From the Epic of Gilgamesh all the way to modern medicine, the trajectory has always been fixed.
One-way street.
A one-way street. You can maybe delay the end, make the decline a little more comfortable with better nutrition or surgery, but you cannot stop the clock.
Yeah.
The consensus has always been that aging is just the accumulation of wear and tear.
The biological equivalent of rust on a car.
That's the perfect analogy.
Right, but has been is the key phrase there, isn't it? Because the research we have in front of us today, it's unsettling in a good way. It suggests that our whole understanding of that law is fundamentally wrong.
It really does. The sources we're looking at are arguing that aging isn't just random entropy. It's not just rust.
It's a biological process.
It's a specific, coordinated set of instructions. And if it's a process, that implies something absolutely radical.
It implies that it can be hacked.
It can be hacked. If aging is a biological program, or at least a condition driven by these mechanisms, then theoretically we can intervene. We can treat it.
And the most provocative claim in all this literature isn't just about slowing it down.
No, not at all. It's about reversing it.
Which, you have to admit, it sounds like alchemy. It sounds like we're back in the 16th century trying to brew the elixir of life in some basement.
I get that. But we aren't talking about Mercury and Eye of Newt here.
No. We are talking about peer-reviewed papers from Harvard, from the Salk Institute, major biotech firms. We're seeing these impossible headlines.
Mice regaining their vision after going blind.
Human skin cells being turned back into stem cells.
Yeah.
And then there's that 2022 clinical trial where human beings actually, measurably, shed years off their biological age.
It's a complete paradigm shift from aging as fate to aging as a disease. And that distinction, that's everything.
Why is that distinction so important?
Because we don't accept cancer or heart disease as fate. We attack them. This research suggests we can now attack the root cause of almost all of them, which is the aging process itself.
So that's what we need to do today. We need to walk through this new science. We need to understand the mechanisms, the real nuts and bolts of how we age, and the specific technologies scientists are now using to try and turn that arrow of time around.
And we have to start with that conceptual change because for the longest time, the wear and tear theory was the only game in town.
Right. The old model was just mechanical.
Purely mechanical. Think of your body like a vintage car. You drive it, the tires wear down, the pistons crack, the chassis rusts. That's entropy.
In that view, medicine is just a mechanic, right?
Yeah.
Replacing parts.
Exactly. You get a hip replacement, that's a new tire. You take a statin, that's cleaning the fuel line. But you can never make the engine new again. You can't unwind the odometer.
Because the damage is physical. It's
Yeah.
permanent.
Yeah.
A shattered glass doesn't just unshatter.
Precisely. But then the research started to get weird. This was in the 1990s, and it really, really accelerated in the 2010s.
What started to change?
Biologists began to notice that cells didn't just wear out. They changed their behavior. It was like they were receiving different instructions.
So it's not the hardware failing, it's a software problem.
That's the core of it. And the breakthrough that really shattered the old model, the smoking gun, if you will, was a study from the Salk Institute in 2016.
This is the one with the progeria mice, the ones that age incredibly fast?
Yes, them and normal mice too. But the core of it was their use of cellular reprogramming. We'll get into the specific factors later, these things called Yamanaka factors, but the concept is what's so important here.
Okay, so what did they do?
They took cells that were biologically old, Their mitochondria were sluggish, their DNA repair was slow, they looked decrepit. And they didn't just patch them up, they pulsed them with these reprogramming factors.
And what happened? I mean, did they just look superficially younger, like putting a new coat of paint on that rusty car?
No, and that's the crucial distinction. If you paint over rust, the car still doesn't run well. These cells started functioning as if they were young.
Functioning how?
Their energy production spiked, their ability to divide was restored, and most importantly, the epigenetic marks, these are the chemical tags on their DNA that accumulate with age, they were wiped clean.
So it wasn't that the hardware was broken after all. The DNA, the hard drive was fine.
The hard drive was perfectly fine. It was the software. The operating system had become corrupted over time.
Which is the information theory of aging. That's David Sinclair's whole idea, right?
It is, a concept championed by him and others. The idea is that we don't age because we run out of resources or because our genes mutate into oblivion. We age because our cells lose their identity.
They forget what they're supposed to be doing.
Exactly. They forget which genes to turn on and which to turn off. The Salk study showed that a backup copy of the original software still exists in the cell. We just needed to find a way to reboot the system to restore it.
That is it's mind-bending. It suggests that inside every 80-year-old body, the blueprint for a 20-year-old body is still there, completely intact, just waiting for the password.
That is exactly what the data implied.
Okay. But before we get too excited about the reboot, we have to understand the crash. If we're going to treat aging like a medical condition, we need a proper diagnosis.
We do.
And the field has moved away from vague terms like getting old and developed a very specific list, the hallmarks of aging.
Right. In 2013, a landmark paper defined these. Originally, there were 9, though that list is always being debated and updated. But to really grasp what's going on, it helps to group them.
It makes it less of a laundry list and more of a narrative of failure.
Exactly. A story of how the system breaks down.
Okay, let's break them down then. The first group seems to be about information failure. This is where the blueprints get messy. It includes genomic instability, telomere attrition, and epigenetic alterations.
Right. So let's start with genomic instability. This is the classic damage model that we've known about for a while.
This is actual damage to the DNA itself.
Yes. Your DNA is a string of 3 billion letters. Every single day, UV radiation from the sun, chemicals in our food, even our own metabolism cause breaks in that string.
And some breaks are worse than others.
The double-strand breaks are the nasty ones. They literally snap the ladder in half.
But we have repair crews, don't we? Enzymes that are supposed to rush in and fix the break?
We do. We have fantastic ones. Proteins like PRRP1 and the sirtuins, they're the first responders. But here's the problem.
They get overwhelmed.
They get completely overwhelmed. As you get older, you have more breaks. The first responders are constantly rushing from one emergency to another. And while they're busy fixing a broken window over here, they aren't doing their day job, which is regulating genes over there.
So distraction leads to chaos. The main work doesn't get done because they're always putting out fires.
Precisely. And that leads right into the telomeres. This is the hallmark most people have probably heard of.
The shoelace analogy, right? The little plastic tips at the end of our chromosomes.
It's a useful analogy, even if it's a bit simplified. Every single time a cell divides, it has to copy all of its DNA. But the copying machine, an enzyme called DNA polymerase, it can't quite reach the very end of the strand.
So a little bit gets chopped off each time.
A little bit gets chopped off. The telomeres are sacrificial buffer zones. They don't code for anything important. They're just there to get shorter with every division.
And what happens when they run out?
The cell hits what's called the Haylick limit. Back in the 1960s, a researcher named Leonard Hayflick discovered that human cells can only divide about 50 times before they just stop.
And that's because the telomeres are gone.
Once the telomeres are critically short, the cell senses the exposed DNA as a major break. It hits the emergency stop button. It either dies or it becomes senescent, which is a huge problem we'll get to in a minute.
But there's an enzyme that fixes this, isn't there? I remember reading about telomerase.
Yes, telomerase can rebuild the caps. It's like retipping the shoelaces. But in most of our adult cells, the gene for telomerase is switched off.
Why? That seems like a design flaw.
It's a trade-off, a terrible trade-off, but a necessary one from an evolutionary perspective. If cells can divide forever, that's a very high risk for cancer.
So evolution decided it was better for us to age than for us to get tumors when we're young.
We age to prevent cancer. Which brings us to the third part of this group, and maybe the most critical one for this new science of reversal, epigenetic alterations.
This is the software you mentioned,
Right.
not the hardware.
This is the software. Your DNA is wrapped around these little spools called histones, and chemical tags, mostly methyl groups, attach directly to the DNA to tell the cell, read this gene, ignore that gene.
This is how a liver cell knows it's a liver cell and not a neuron, even though they have the exact same DNA.
Exactly. But over time, because of those distracted repair crews we talked about, these tags get shuffled. The wrong ones get put on, the right ones fall off.
The methylation patterns drift.
It's called epigenetic drift. And the result is chaos. A skin cell starts trying to express a few liver genes. A neuron starts expressing kidney genes. The cell loses its identity. It becomes a generic, confused, low-functioning cell.
It's noise. The noise grounds out the signal of youth.
That's the perfect way to put it.
Okay, so that's group 1. The instructions get garbled. Now, group 2 is what the literature calls compromised maintenance. Basically, the housekeeping falls apart. This covers things like loss of proteostasis and mitochondrial dysfunction.
Right. So let's start with proteostasis. It's just a fancy word for protein stability. Your cells are fundamentally protein factories.
But proteins are like complex origami, right? They have to be folded perfectly to work.
Exactly. If they misfold, they're useless at best. At worst, they're sticky.
Sticky, what do you mean?
They clump together into aggregates. Usually we have a system called the ubiquitin proteasome system that tags these bad proteins and shreds them. There's also a process called autophagy, which is basically the cell eating its own garbage.
The cellular recycling system.
Correct. But as we age, both of these systems get sluggish. The garbage bin overflows.
And these clumps of sticky, misfolded proteins. These are the plaques we see in diseases like Alzheimer's, aren't they? The amyloid beta.
Precisely. Alzheimer's, Parkinson's, Huntington's, these are at their core diseases of failed proteostasis. The neuron is literally choking on its own waste.
And while the house is filling with trash, the power starts to go out. Mitochondrial dysfunction.
Because mitochondria, everyone remembers from high school biology, they're the power plants of the cell, generating ATP.
But they have their own DNA, which is a problem.
A huge problem. It's not protected inside the nucleus, so it's very susceptible to damage. As the mitochondria get older, the electron transport chain, the machinery that actually makes the energy, starts to leak.
It's leaking electrons.
Yes, and those leaked electrons react with oxygen to form free radicals, or reactive oxygen species. These things are incredibly toxic. They're like little grenades going off inside the cell, damaging everything they touch.
So an old mitochondrion is producing less energy and more pollution.
It's a double whammy. Less power for the cell to function, and more damage causing even more dysfunction.
So we've got bad instructions, a house full of trash, and a power plant that's spewing smoke. This brings us to the third group, systemic failure. This is where the damage stops being a local problem and spreads to the whole neighborhood.
And the star of this show is cellular senescence.
Zombie cells. I love that the scientific community actually embraced that term.
It fits perfectly. A senescent cell is a cell that has suffered too much DNA damage, or its telomeres have gotten too short. It stops dividing, which is good. It should then die, a process called apoptosis, or programmed cell death.
But it doesn't.
It doesn't. It refuses to die. It enters this bizarre state of suspended animation.
But it's not passive, is it? It's not just sitting there.
Far from it. It becomes hyperactive in a very bad way. It develops what's called the SASP, the senescence-associated secretory phenotype.
That's a mouthful.
It is. But what it means is that the zombie cell basically starts vomiting out a toxic cocktail of inflammatory cytokines, growth factors, and enzymes that degrade the tissue around it.
So it's a toxic neighbor. It's not just messy. It's actively damaging the property of everyone else on the block.
Worse, it's recruiting. The inflammation it spews can actually cause its healthy neighbors to turn into zombie cells, too.
So it spreads.
It spreads. And this chronic, low-grade inflammation that results, scientists now call it inflammaging, is a primary driver of almost every age-related disease you can name, from arthritis to heart disease to fibrosis.
And I'm guessing our immune system, which is supposed to clear these things out, is also getting old and tired.
Exactly. The police force is understaffed and getting slower, and the rioters are multiplying and recruiting others to their cause. It's a recipe for disaster.
Wow. Okay, this paints a pretty grim, cascading picture of total system failure. But the reason we're here, the reason this research is so exciting, is that by identifying these specific hallmarks, we now have targets.
That is the pivot point. We've moved from just observing and asking, why is this happening, to actively intervening and asking, what can we do about it?
If we know it's a zombie cell problem, we can try to kill the zombies. If it's an epigenetic noise problem, we can try to retune the radio.
And the interventions being developed are just fascinating.
Let's start with the biggest one, the one that arguably kicked off this entire reversal craze: cellular reprogramming.
This goes back to a discovery by Shinya Yamanaka in 2006, for which he won the Nobel Prize.
Right. What was his big question?
He asked a deceptively simple question. How does a fertilized egg, which comes from 2 adult cells, an egg and a sperm, reset the clock to 0 to create a brand new baby?
How does nature do it?
Exactly. And he found 4 specific transcription factors. These are proteins that bind to DNA and tell it which genes to express, that can take any adult cell, like a skin cell, and turn it all the way back into a pristine, embryonic-like stem cell.
The Yamanaka factors. We should name them for the record.
They are OCT04, SOX2, KLF4, and C-Mike. Together, they're known as OSKM.
And if you express these genes in a skin cell, you say it erases the epigenetic identity completely.
Completely. It becomes what's called an induced pluripotent stem cell,
Right.
or iPSC. It can become any cell in the body.
Which is amazing for growing tissues in a petri dish. But if you do that in a living animal,
It's a catastrophe, a total disaster. If you wipe the identity of a liver cell while it's still sitting in the liver, it stops doing liver things. It just becomes a blob.
that's the best case scenario.
Yes. Even worse, one of the factors, C-Mike, is a potent oncogene. It drives cancer. The early experiments with continuous reprogramming resulted in mice that were just riddled with teratomas.
Teratomas are those horrible tumors with teeth and hair in them.
Yes, tumors made of mixed-up tissue, like teeth and hair growing inside the kidneys. It's nightmare fuel.
So how on earth do we use this for aging without turning into a tumor monster?
This is where the idea of partial reprogramming comes in. The Harvard group in David Sinclair's lab had a brilliant idea.
What if you don't push the reset button all the way down?
Exactly. What if you just tap it? The idea is to turn the factors on for just a short burst, just enough to wipe away the epigenetic noise of aging, but not long enough to erase the cell's identity.
And they tested this.
They did. In that famous 2020 Nature paper on the mice eyes, they modified the cocktail.
They took out the most dangerous ingredient.
Right. They dropped C-Mike. They just used OCT4, SOX2, and KLF4, the OSK cocktail. And they used a harmless virus to deliver it to the retinal ganglion cells of old blind mice.
And these mice had damage similar to glaucoma.
Yes, either from a crushed optic nerve or high intraocular pressure. And the result was staggering.
They could see again.
They could see again. But it's more than that. The nerves actually grew back. The DNA methylation age of the cells, according to the epigenetic clock, reversed. The cells accessed that backup copy of youth we talked about.
It proved you could decouple cellular identity from cellular age.
That's the holy grail right there. You can make a cell young without making it forget what it is.
That is incredible, but it's also gene therapy. It involves viruses. It's going to be complex and expensive for a long time. There's a more chemical approach. Let's talk about senolytics.
This is all about targeting the zombie cell.
Killing the zombies.
This is much closer to traditional pharmacology. The idea is, if senescent cells are the problem, let's design a drug that selectively kills them but leaves healthy cells alone.
How do you do that? How does the drug know which cell is a zombie?
It turns out, to avoid dying, zombie cells have to rely on specific survival pathways. They're constantly holding up a shield to stop themselves from committing suicide or apoptosis. Most of these pathways involve the BCL2 family of proteins.
So senolytics are drugs that knock that shield down.
Exactly. They inhibit those pro-survival networks. And the most famous senolytic combo is a strange one, dasatinib and quercetin.
Dasatinib. That's a chemotherapy drug, isn't it? For leukemia.
It is, a very heavy hitter. And quercetin is a flavonoid, a natural compound you find in things like capers, onions, and apples.
A chemo drug and a salad ingredient. That is a weird cocktail.
It is, but it works because they target different types of zombie cells in different tissues. And in mice, the results have been phenomenal.
What do they see?
In some studies, clearing these cells extended lifespan by up to 36%. But more importantly, it extended health span. The old mice could run further on their treadmills, their fur got shinier, their heart and kidney function improved dramatically.
And this is actually in human trials now, isn't it?
Yes, there are a number of trials for specific age-related conditions like idiopathic pulmonary fibrosis and diabetic kidney disease. The Mayo Clinic is a big leader here.
So the idea isn't a live forever pill just yet, but a targeted treatment for an organ that's failing because it's full of zombie cells.
That's the first step, and it's a huge one.
Okay, now the third approach. This one requires a bit of a history lesson, and it's a little bit creepy. It's the young blood research. Parabiosis.
It sounds like something from a Gothic novel, but it's real science. It actually dates back to the 19th century. A French physiologist named Paul Bert was the first to literally sew two rats together in the 1860s.
Creating a shared circulatory system.
Yes. It was grotesque, but it proved that factors in the blood could transport physiological signals from one animal to another.
But the modern interest really reignited in the 2000s, right, at Stanford?
Yes. In the lab of Irina and Michael Conboy, they refined the technique and connected a young mouse and an old mouse. The results were startling.
What happened to the old mouse?
Its muscles regenerated as if it were young. Its heart function improved. New neurons started growing in the hippocampus, the memory center of the brain. It healed faster from injury.
And the young mouse? What happened to it?
It suffered. It aged rapidly. Its tissues showed signs of damage and inflammation.
Which sparked this huge debate in the field. Is it that young blood has good stuff in it, or does old blood have bad stuff that needs to be diluted?
The elixir versus the poison debate. Exactly. For a while, some researchers isolated a protein called GDF11 and claimed it was the key youth factor. Other labs couldn't replicate it. The current consensus is that it's probably a mix of both.
Old blood is full of that inflammatory SASP from the zombie cells.
Right. And young blood has optimal levels of various growth and repair factors.
There was that company, Ambrosia, a few years back that started selling young plasma transfusions to wealthy people in Silicon Valley.
There was, and the FDA shut them down pretty hard.
And rightfully so.
Absolutely. There was no clinical evidence it worked in humans, and the risks of transfusion reactions are very real. The real science is moving toward identifying the specific proteins. We don't want to be hooking people up to each other. We want a synthetic serum that contains just the beneficial active factors.
That feels a lot safer. Okay, so approach 4 is metabolic hacking. This is about fueling the machine properly and cleaning it out.
This is an area with a lot of excitement. We talked about mitochondria failing. Well, one of the key components for their function is a molecule called NAD+I.
Nicotinamide adenine dinucleotide.
That's the one. It's a coenzyme in all living cells. It's essential for converting energy from food into cellular energy, ATP, but it has another critical job.
It fuels the repair crews.
It's the fuel for the sirtuins, those DNA repair proteins. Sirtuins are NAD dependent. They can't work without it.
And I'm guessing our NAD levels drop as we age.
They plummet. By the time you're 50, you might have half the NAD levels you had when you were 20. So the repair crew literally runs out of gas.
So the logic is, if we refill the tank, the repair crew gets back to work. And that's why people are taking precursors like NMN or NR.
Yes, nicotinamide mononucleotide and nicotinamide riboside. The research in mice is very clear. Feeding these to old mice restores their NAD levels and rejuvenates muscle, brain, and other tissues. The human data is still coming in, but it's one of the most popular interventions because they're widely available as supplements.
And then there's rapamycin. This one is different because it's not a supplement, it's a serious drug.
Rapamycin is fascinating. It was discovered in a soil sample from Easter Island, Rapa Nui, hence the name. It's a powerful immunosuppressant used to prevent organ rejection in transplant patients.
But it has this incredible side effect.
It turns out to be the most potent inhibitor of a protein called mTOR.
And mTOR is like the master growth switch in the cell.
Think of mTOR as the construction foreman on a job site. When mTOR is on, the cell is in growth mode. It's building, it's dividing, it's using resources. When mTOR is off, the foreman tells the crew to stop building and instead clean up the site and repair the tools.
So it activates autophagy, the cellular cleanup process.
It's a powerful activator of autophagy. So blocking mTOR with rapamycin basically tricks the cell into thinking times are tough, that it's starving. It forces it into a survival and repair mode.
And this works?
Rapamycin is the most robust and reproducible pharmacological intervention for extending life in mice. It works in yeast, worms, flies, mice. It's incredibly consistent across species.
But it suppresses the immune system. That's a pretty big risk for a longevity drug you'd take for decades.
It is the major hurdle. That's why a lot of research is focused on intermittent dosing, maybe once a week, or on developing rapalogs, which are new drugs that mimic the life extension benefits without the heavy immune suppression.
So we have all these amazing interventions, reprogramming, zombie killing, young blood factors, metabolic tweaks. But here's the problem. Humans live a long time.
They do.
If I take a pill today, I don't want to wait 40 years to see if I die later than my neighbor. We need a speedometer. We need to know if any of this is working now.
This was the bottleneck of the entire field for decades. until Steve Horvath came along.
The Horvath clock. The epigenetic clock.
In 2013, Horvath, who's a biostatistician at UCLA, had a breakthrough. He realized that those methylation patterns on our DNA, the epigenetic tags we talked about, they don't just drift randomly. They change in a highly predictable way as we age.
So he found specific spots on the genome that are like a timestamp.
Exactly. He identified 353 specific sites, CPG islands, where methylation reliably increases or decreases with age. He built an algorithm that could look at the methylation status of just those spots from a drop of blood and tell you your age to within a few years.
But it's more than just telling you your birthday. It tells you your biological age.
That's the key. If you're 50 chronologically, but you've smoked your whole life and have been under high stress, your methylation patterns might look like a 60-year-old's. You are biologically aging faster than the calendar.
This gave scientists a ruler, a way to measure aging in real time.
It changed everything. Suddenly, you could run a clinical trial for 1 year, not 40 years, and see if your intervention made people biologically younger.
And that ruler was used in the first study that suggested human age reversal is actually possible. The TRIUMPH trial.
The TRIUMPH trial. It stands for thymus regeneration, immunorestoration, and insulin mitigation. It was a small study led by a scientist named Greg Fahey.
And he was targeting the thymus gland.
Right. The thymus is this little gland that sits behind your breastbone. It's where your immune T cells mature. It's crucial for a young, adaptive immune system. But it starts to shrink after puberty, and it's basically turned into useless fat by the time you're 50 or 60.
Fahey wanted to regrow it.
He did. He used a cocktail of drugs. The main one was human growth hormone, or HGH, which is known to regenerate tissue.
But HGH has a dark side. It can increase cancer risk, and it spikes insulin, which accelerates aging.
Correct. So to counteract that, he combined it with 2 other drugs. Metformin, the diabetes drug to control insulin, and DHEA, a steroid hormone.
And the results were published in 2019. What did they find?
They were stunning. But we have to add a huge grain of salt here. The study only had 9 participants. They were all white men aged 51 to 65. And there was no control group.
Scientifically, that's a very weak structure for a trial.
Very weak. It's more of an observation than a rigorous experiment. But the clock data.
The clock data was undeniable.
It was. On average, after 1 year of treatment, the participants had lost 2.5 years of biological age, according to the Horvath clock. Their thymus glands showed signs of regrowth on MRI scans. Their immune profiles looked younger and healthier.
So even with the tiny sample size and no controls, the fact that the arrow moved backward at all is historic.
It is. It's the proof of principle. It showed that the epigenetic clock isn't just a one-way counter. It can, in fact, be wound back in humans.
Okay. So we have a theory, we have the tools, and we have a small proof of concept. So why aren't we all immortal yet? We have to talk about the catch. The challenges section of our notes is pretty substantial.
And the biggest challenge, without a doubt, is the Goldilocks problem of cancer.
Explain that. What is the Goldilocks problem?
Evolution is a delicate balance. We have tumor suppressor genes for a reason. Cellular senescence, those zombie cells, didn't evolve by accident. They evolved as a powerful defense mechanism.
To stop a damaged cell from becoming cancerous.
Exactly. The body's response to a cell that is at risk of becoming cancerous is to shut it down permanently. So if we come in with our fancy drugs and force those cells to wake up, or if we use telomerase to make our telomeres longer,
we might just be disabling the safety brakes on a car that's speeding down a hill toward a cliff.
That's the fear. Telomerase, the enzyme that lengthens telomeres, is active in 90% of all human cancers. It's what allows tumors to achieve immortality and grow indefinitely. If we turn on telomerase everywhere in the body, we might get longer life, or we might just get aggressive system-wide cancer.
And the reprogramming factors, you said c-Myc is a known oncogene.
A powerful one. Even without c-Myc, tampering with a cell's identity is incredibly risky. We need precise, reliable control. We need to be able to turn these factors on and off with absolute certainty. We are not there yet.
And then there's the simple fact that humans are not 70-gram mice. We've cured cancer in mice 1,000 times over.
It's true. Humans are large, long-lived animals. Our metabolic rates are different. We're exposed to a lifetime of different environmental toxins. A mouse lives 2 years. Its repair mechanisms are tuned for a short, fast life. Translating mouse success to human success has historically been the graveyard of biotech.
There's also a huge ethical and societal angle here. If these treatments work, they will not be cheap.
Not at all. We're talking about complicated gene therapies, personalized stem cell treatments. This isn't going to be like buying aspirin at the pharmacy.
No. And that creates the very real risk of biological caste system.
A longevity dividend for the rich. Imagine a world where the wealthy elite can afford to reset their epigenetic clocks every decade. They stay young, healthy, and productive for 150 years. Meanwhile, the rest of the population ages and dies normally.
It would amplify every existing inequality to an unimaginable degree. Wealth already buys better health, but this would mean wealth buys life itself. The compounding interest of living an extra century would make current wealth gaps seem tiny.
It's a geopolitical nightmare. And what about social structures? What happens to retirement? If you live to 150 in good health, you can't retire at 65, the pension systems would instantly collapse.
You'd have to work until you're 120.
At least. And what would that do to the job market? It would be completely stagnant. If the CEO never ages and never retires, how does a 30-year-old ever get a promotion?
Innovation often comes from the turnover of generations. As a physicist Max Planck famously said, science advances one funeral at a time.
If the old guard never leaves, does society stagnate? Do we lose our capacity for new ideas?
These are incredibly heavy questions. But let's try to bring it back to the present for a minute. The listener's probably thinking, okay, society might collapse in 50 years, but I want to have more energy today. What is the practical takeaway for the here and now?
Well, the good news is that the research strongly suggests that while we wait for the gene therapies, the boring stuff works via many of the same pathways.
You're going to say exercise, aren't you?
I have to. It's unavoidable. High-intensity interval training, HIAT, has been shown to boost mitochondrial function better than almost anything. It increases NAD+ levels. It stimulates autophagy. It is, functionally, a longevity drug with only positive side effects.
What about diet? Fasting?
Caloric restriction is the only intervention that has reliably extended lifespan in almost every species ever tested, from yeast to monkeys. Now, you don't have to starve yourself, but things like time-restricted feeding, just giving your body a 14 or 16-hour break from digestion each day, that activates those same repair crews.
So skipping breakfast is basically a weak, free version of taking rapamycin.
Biologically, yes. You're turning down mTOR and turning up the sirtuins and autophagy. They hit the same buttons.
Okay, let's look at the horizon, the future.
Yeah.
Where is all this going in the next, say, 5 to 10 years?
I think in the next decade, we are going to see the approval of the first true longevity drugs. They won't be marketed that way, of course.
They'll be for specific diseases.
Exactly. I'd bet on senolytics first, targeting specific diseases like kidney failure or lung fibrosis. But once a drug is approved for a disease, doctors can prescribe it off-label, and people will absolutely use it for general aging.
And beyond that, the radical future. What about 20, 30 years from now?
That's where you get into the concept of longevity escape velocity.
This is Aubrey de Grey's idea. He's the biomedical gerontologist.
He is. He's the one who popularized this. The idea is that science and technology are accelerating.
Right now, for every year you live, science might add, what, a few weeks or a month to your total life expectancy?
Something like that. But eventually, as AI, biotech, and nanotechnology converge, we might reach a tipping point. A point where for every year you manage to stay alive, science is able to add more than a year to your remaining life.
You literally outrun the Grim Reaper.
Theoretically. The idea is if you can just survive the next 20 or 30 years, you might be able to survive for 1,000. If you can continually repair damage faster than it accumulates, there is no longer a biological limit on your lifespan.
That changes the very definition of what it means to be human. We are defined by our mortality. Our art, our poetry, our religions, our sense of urgency, it all comes from the knowledge that our time is limited.
It does. If you have 500 years, do you still try to write that novel today? Do you marry that person this year, or do you just drift?
Yeah, I'll get to it next century.
The psychology of radical life extension might be the hardest hurdle of all.
It's incredible. We've gone from looking at aging as an immutable law of physics to looking at it as a software bug. We've identified the code, the hallmarks. We're building the tools to fix it. Reprogramming senolytics. We have the metrics to measure our progress, the clock. It really feels like we are standing on the edge of a cliff, waiting to see if we can actually fly.
The door is open. The science has fundamentally moved from slowing the decline to reversing the damage. Now, it's an engineering problem. A very, very complex one, but an engineering problem nonetheless.
I'll leave everyone with a thought experiment. Because it's one thing to talk about this with mice, but it's another to think about it for yourself. So imagine it's 10 years from now, it's 2036. The FDA approves the first real age reversal treatment, a gene therapy shot. It promises to lock your biological age right where it is today. You won't get a day older biologically, but there's a catch. Because it involves permanently altering your genes, there's a known 5% chance it will cause a fatal, untreatable cancer within 20 years. Do you take the shot? Do you gamble a natural death in your 80s or 90s for a shot at practical eternity?
That's a tough one.
I think a lot of us would hesitate a lot more than we might admit right now.
I think I'd wait for the version 2.0 update, the one with the bug fixes.
Always the cautious scientist. Thank you so much for walking us through this incredible, world-changing material.
It was my pleasure.
And to you, listening, thanks for joining us. Keep your mitochondria happy, maybe skip breakfast tomorrow, get some sleep, and we'll see you next time.